Welcome to our lesson on culturing microorganisms. The key questions for today are: what process do bacteria use to reproduce? What can affect the rate of bacterial reproduction? How can we use cultured bacteria colonies to test disinfectants and antibiotics? How can you prepare an uncontaminated culture of bacteria in a school laboratory? Why should school-grown bacteria be incubated at 20 degrees centigrade? Key terms that you will need to know are binary fission, agar gel, aseptic, antibiotic, incubation. We already know that bacterial cells are prokaryotic cells that have a loop of free DNA in the cytoplasm. They also contain plasmids that are extra sections of DNA. Harmless bacteria are present on lots of surfaces and even in the body, but some types can cause infections. This is why we need to be able to find out about how quickly they reproduce and what can kill them. We can do this by culturing bacteria. This means introducing a small sample of bacteria to a food source such as agar and allowing it to reproduce to form colonies that are visible. We can measure the area of the colonies to estimate the number of bacterial cells present. Bacterial cells reproduce by a process called binary fission. This process sees one bacterium divide to form two new bacterial cells. The rate at which this division happens can allow us to work out how many bacteria will be present after a set amount of time. If you begin with one bacterial cell and binary fission takes place every 20 minutes, after 1 hour there would have been 3 divisions. In the first division, the original cell splits into 2. In the second, those 2 split to make 4. Then in the third division, those 4 split to make 8. This means that after 1 hour, there would be 8 bacterial cells. Task: if you begin with 5 bacterial cells that undergo binary fission every 30 minutes, how many cells would you have after 2 hours? In 2 hours, there would be 4 divisions, so there would be 80 bacterial cells after 2 hours. After each division, the number of bacteria doubles. So 5 to begin with, 10 after the first division, 20 after the second, 40 after the third, and 80 after the fourth. Extension. Complete the following AQA exam question. A bacterial colony will grow at a steady rate providing there is enough food, the temperature is optimal, and there are no toxins present. If a colony runs out of the food source, the colony will stop growing, and the number of live bacteria present will start to decrease as many will begin to die because of lack of food. A bacterial colony with enough food may appear to stop growing, as the number of bacterial cells dying will be the same as the number of new cells produced. Temperature can affect the rate a colony grows. If it's cold, then reproduction will be slower. At warmer temperatures, the bacteria will reproduce more quickly. Other factors, such as pH, can also affect bacterial growth, with all bacteria having an optimum or preferred pH. When the pH is at this value, the bacteria will reproduce the quickest. When culturing bacteria at school, you must use aseptic technique. This means that no contaminants will be present in your bacteria colony. To grow bacteria, you must first prepare a sterile food source for them to grow on. The food source is agar jelly, and it is heated to a high temperature to kill any bacteria before being set into Petri dishes. These Petri dishes have lids placed over them to stop any contaminants in the air falling onto the agar. The next step is to sterilize your inoculating loop, a small metal loop of wire used to transfer your bacteria onto the agar. You can do this by heating it in a blue Bunsen burner flame for a few seconds. You then need to dip your inoculation loop into the bacteria sample and streak it across your agar. Lift the lid on the Petri dish for the shortest time possible to prevent any microbes from the air entering. Next, seal the lid onto your Petri dish with 2 short lengths of tape. But make sure you leave some gaps for oxygen to enter. You will then need to incubate the sample to allow the bacteria colony to grow. In schools, bacteria is incubated at about 20 degrees centigrade. This is below human body temperature. This is so that no bacteria that could be harmful to us is grown. Task: explain why the inoculation loop is heated in a flame. Extension: why should the inoculation loop be allowed to cool before dipping it into the bacteria sample? The inoculation loop is heated to kill any bacteria that may be present on it. It should not be dipped into the bacteria for the culture until it has cooled, as it would kill the bacteria intended to be grown. Bacterial cultures can be used to test the effectiveness of antibiotics or disinfectants. Small discs of paper are soaked in the substance and placed on the agar. Bacteria will not grow around the discs if they are effective at killing them. The largest clear area indicates the most effective substance. You can work out the area of the clear area using the area of a circle formula. Area of a circle equals A equals pi r squared. You would need to measure the diameter of the clear area and divide it by 2 to find the radius. Task: which letter indicates the most effective antibiotic? D indicates the most effective antibiotic, as it has the largest clear space around it where no bacteria is growing. The bacteria will not grow where there is an effective amount of antibiotic present. The antibiotic will diffuse through the agar and stop the bacteria growing. If bacteria was growing there already, it would kill it, clearing the area. Extension: how could a scientist test the effectiveness of kitchen cleaning fluids on bacteria? A scientist could prepare a culture of bacteria using aseptic technique, adding discs of paper soaked in the cleaning fluids to the surface of the agar. The culture should then be incubated and allowed to grow. The scientist would need to see the clear areas around each disc. The disc with the largest clear area would be the most effective. The diameter of the clear areas could be measured with a ruler and the radius and then the area calculated. Other factors that may need to be taken into consideration would be cost and suitability of the cleaner for a kitchen. For example, if it is poisonous or bad smelling. If the cleaner was poisonous to humans, it would not be suitable to be used around food. If it was bad smelling, it would not be ideal to use in a restaurant.